Hello students, good morning. My name is Ushasri. Today I am going to explain you about biochemical monitoring of athletes during sports training. In this regard, in my previous video, I have covered about nutritional markers and metabolic markers. Today I'll explain about muscle damage markers and overtraining markers. Let's talk about muscle damage markers. Muscle damage markers are the biochemical parameters that can be tested to detect muscle damage in athletes. This group of parameters include serum enzymes, namely creatine kinase and lactate dehydrogenase, myoglobin, and myosin heavy chain fragments. Exercise-induced muscle damage is very common among the athletes. Due to this, some of the enzymes which are present inside the muscles like CPK and lactate dehydrogenase will be released into the blood. So, in the blood, these enzyme concentrations increases. These two diagrams shows the CPK activity and LDH activity before and after the exercise. If you see this graph, in both the cases, post-exercise values are higher than the pre-exercise values. This means muscle damage is common to some extent in athletes, but severe muscle damage may result in further increase in these enzyme levels. So, CPK and lactate dehydrogenase can be used to diagnose muscle damage and to understand the extent of muscle damage. These parameters also can be used to monitor the recovery of athletes after muscle injury. Muscle activity requires continuous supply of ATP. We have different mechanisms for the regeneration of ATP. One of such mechanism is phosphagen system. In phosphagen system, ATP will be produced from ADP when the creatine phosphate donates its phosphate group to ADP. This reaction is catalyzed by an enzyme called creatine phosphokinase. This is a reversible reaction. It can occur in both the directions. When the ATP levels are low, creatine phosphate donates its phosphate group to ADP and produces ATP and creatine. When ATP levels are high, phosphate group is donated from ATP to creatine in order to produce phosphocreatine. Creatine phosphokinase is also referred as creatine kinase. CPK has 3 isoenzymes. What are isoenzymes? Isoenzymes are the enzymes which differ in their structure but they catalyze same reaction. So CPK has 3 isoenzymes, namely CKMM, CKMB and CKBB. CKMM is mainly present in muscles, skeletal muscles. CKMB is mainly present in cardiac muscle and CKBB is mainly present in the brain. So any skeletal muscle injury would result in increased levels of CKMM in the blood. Any cardiac muscle injury would result in increased levels of CKMB in the blood, and any injury related to the brain would result in increased levels of CKBB in the blood. So when we are measuring CPK, we should make sure the increased CK is not CKMB or CKBB, which is only CKMM, in order to diagnose skeletal muscle injury. Lactate dehydrogenase is an enzyme involved in energy metabolism. It catalyzes the interconversion of pyruvate and lactate. When oxygen is absent, pyruvate will be converted to lactate. It is a reversible reaction. Same enzyme can convert lactate to pyruvate also with the interconversion of NAD to NADH. Lactate dehydrogenase has 5 isoenzymes. All the 5 isoenzymes are tetramers, means they are made up of 4 subunits of either H type or M type or a combination of it. If you see this LDH1 isoenzyme, it is mainly made up of 4 H subunits. LDH2 is made up of 3 H subunits and 1 M subunit. LDH3 is made up of 2 H subunits and 2 M subunits. LDH4 is made up of 1 H subunit and 3 M subunits. LDH5 is mainly made up of 4 M subunits. Among these 5, LDH4 and LDH5 are more specific to skeletal muscle. So more predominantly present in skeletal muscle. So any skeletal muscle injury would result in increased levels of LDH4 and LDH5 in the blood. On the other hand, any cardiac injury results in increased levels of LDH1. If the blood sample is lysed, if the RBC are lysed, that will lead to increased levels of LDH2. So utmost care need to be taken while estimating the LDH levels, because any lysis of RBC can give increased LDH levels in the blood, and that can be falsely interpreted as skeletal muscle injury. So lysed samples should not be taken for LDH estimation. Myoglobin is an iron-containing protein present in muscle fibers. It serves as the intracellular storage site for oxygen in the muscles. Strenuous exercise can create periods of O2 deprivation. Then the oxymyoglobin releases its bound oxygen for metabolic purposes. Extensive damage to the skeletal muscles results in the excretion of myoglobin in the urine. This is called myoglobinuria. This can be observed in rhabdomyolysis. Rhabdomyolysis means the breakdown of striated muscles that include skeletal muscle as well as cardiac muscle. So myoglobin levels increases even in cardiac muscle injury. After skeletal muscle injury, within 2 to 6 hours, the blood levels of myoglobin increases, and usually myoglobin levels above 85 Ng per ml can be observed in conditions such as rhabdomyolysis, muscle inflammation, and muscular dystrophy. Myosin heavy chain fragment is a structurally bound contractile protein of thick filaments. Increase in plasma MHC indicate both membrane leakage and degradation of contractile apparatus. Usually MHC levels increases after 48 hours of muscle injury, and the measurement of MHC will be useful in the diagnosis of previous rhabdomyolysis up to 12 days. So myoglobin is the quick marker to identify muscle damage, means within 4 to 6 hours we can identify the muscle damage by estimating myoglobin levels. However, estimation of serum enzymes and myosin heavy chain fragments will be helpful in the delayed diagnosis of muscle damage. Means after few days, if you want to diagnose whether muscle damage has happened or not, we can choose these parameters. Now let's check out the biochemical markers of overtraining. Constant intense training that does not provide adequate time for recovery may lead to overtraining. Overtraining results in sustained reductions in performance of the athletes and is often accompanied by other biochemical, physiological, and psychological changes. Parameters that are tested to know the overtraining and recovery status of an athlete include cortisol, testosterone, testosterone cortisol ratio, sex hormone binding globulin, glutamine glutamate ratio, creatine phosphokinase, lactate response, and C-reactive protein, etc. Cortisol is a catabolic hormone produced from adrenal cortex. Cortisol secretion is stimulated by stress. Exercise or any form of stress results in the increased production of corticotropin releasing hormone from the hypothalamus. This in turn acts on anterior pituitary gland and results in the increased release of adrenocorticotropic hormone, and it acts on adrenal cortex and results in the secretion of cortisol. Exercise induced hypoglycemia, that is decreased levels of blood glucose and increased physical stress during the exercise, results in the increased production of cortisol. Main function of cortisol is to maintain blood glucose levels. How cortisol maintains the blood glucose levels? Cortisol acts on adipocytes or adipose tissues and increases the lipolysis, that is breakdown of triglycerides into free fatty acids and glycerol. These free fatty acids, which are released into the blood circulation, will be utilized for energy production in other tissues. So mainly energy will be coming from fats instead of glucose. So glucose will be spared. On the other hand, cortisol acts on muscle protein and increases the catabolism of muscle protein and results in the formation of amino acids. And these amino acids are again utilized for the resynthesis of glucose by a process called gluconeogenesis. So this glucose will be again added to the blood circulation. This is how cortisol maintains the glucose levels in our body. Cortisol also blocks the entry of the glucose into the tissues. So altogether, glucose levels in the blood increases and energy production from the fats increases under the influence of the cortisol. At the same time, high levels of cortisol will lead to excessive protein catabolism, and this may lead to loss of muscle protein. When cortisol levels are high, the muscle protein catabolism increases. So for an athlete, it is very difficult to maintain the muscle mass. This may affect their performance negatively. So high cortisol levels are considered as the marker of overtraining in athletes. Consumption of high quality protein will help to decrease the cortisol production, and consumption of low glycemic carbohydrates immediately after the exercise also will be helpful to reduce the cortisol levels. On the other hand, omega-3 fats from fish and flaxseed oil also can help to control cortisol surge. Testosterone is an anabolic hormone secreted by the interstitial cells of testes. It is mainly concerned with the development of male secondary sexual characteristics, and it also promotes masculine characteristics in men. Testosterone is required for promoting protein synthesis, red blood cell production, and glycogen replenishment. On the other hand, optimum testosterone will reduce protein breakdown in athletes. So, low testosterone in athletes may lead to decreased performance, energy levels, and strength. Consumption of good amount of fat, consumption of test boosting ingredients like testofen, that is the extract of fenugreek, consumption of more zinc and vitamin D and D-aspartic acid will help to improve the testosterone levels. Apart from that, good sleep and multi-joint exercises also will be helpful to maintain optimum testosterone levels. Testosterone cortisol ratio will provide a relative indication of anabolic and catabolic balance, especially in male athletes. And compared to the individual testing of testosterone or cortisol, TC ratio is more sensitive and gives valuable information related to the training stress. A prolonged decrease in TC ratio is associated with the detriments to performance through decreased protein synthesis and increased protein catabolism. A 30% decrease in TC ratio has been suggested as an indicator of insufficient recovery, whereas a value of 0.35 into 10 to the power of minus 3 has been considered to be the threshold of overtraining. SHBG is a protein produced primarily in the liver, and this protein transports sex hormones including testosterone throughout the body. In our body, major amount of testosterone is bound to SHBG. Very less amount is in free form. Bioavailability of testosterone is influenced by the level of SHBG. When SHBG levels increases, very less amount of testosterone is left free. So, SHBG is a useful indicator of training status and performance. Increased SHBG and decreased testosterone may indicate insufficient recovery in athletes. Glutamine is an amino acid involved in neural plasticity and protein synthesis. Decreased levels of glutamine may reflect fatigue or suboptimal training adaptation in athletes. Decreased glutamine concentration and increased glutamate concentration results in decreased glutamine-glutamate ratio, and these parameters are widely used as a markers of Overtraining in the field of sports. Besides the above mentioned parameters, the neutrophil lymphocyte ratio or NL ratio also can provide a good measure of exercise stress and subsequent recovery. The NL ratio usually returns to the normal within 6 to 9 hours after the exercise, but where the exercise has been particularly prolonged and stressful, the NL ratio may take almost 24 hours to reach back to the normal range, or it may still be elevated even after 24 hours of post exercise. Along with the above set parameters, the estimation of creatine kinase activity will give information about exercise induced muscle damage and recovery in athletes. Overtraining may lead to decreased glycogen storage in the muscles. Some studies have reported lower blood lactate responses during submaximal exercise test in overtrained athletes. This has been explained on the basis of low muscle glycogen levels and decreased catecholamine responses to exercise in overreached athletes. Nowadays, C-reactive protein is used as a strong indicator of inflammation resulting from tissue injury that is associated with overtraining syndrome and poor recovery. So estimation of CRP also will give some information about inflammation that is initiated from overtraining syndrome.
Thank you.